Today's focus is on one of the most sensitive tools in our arsenal for measuring small molecules, the competitive ELISA. You might have heard of ELISA before. It stands for Enzyme-Linked Immunosorbent Assay. The most common form of ELISA is the sandwich method, where we trap big molecule between two antibodies. But today we're doing something different. We are looking at a tiny signaling molecules called cAMP. Because it is so small, we can't sandwich it. Instead, we have to make it compete in a race. I'm going to walk you through the exact protocol using footage from an experiment I ran just this week, which we will analyze right here on the digital board. I am utilizing this method for my doctoral dissertation at Charles University. My broad research topic is homocysteine, specifically its interaction with physiological metals and its cardiovascular relevance. However, a key part of that puzzle involves understanding cellular signaling. To do that, I need to measure cAMP in blood platelets very precisely. Let's zoom in on the plastic well of our ELISA plate. The bottom of this well is pre-coated with specialized protein called a goat anti-rabbit antibody. Now you might stop and ask, why a goat? And why is it anti-rabbit? This is actually a fascinating story of immunology. To get this molecule, scientists take antibodies from the rabbit and inject them into a goat. The goat immune system sees these rabbit proteins as foreign invaders, as viruses, and produces its own antibodies to attack them. So we harvest these goat antibodies. Their only job in life is to hunt down and grab onto anything that comes from a rabbit. In our well, these goat antibodies are chemically bonded to the plastic floor. Think of them as the anchors. They are stuck to the bottom by their tails, with their grabbing arms waving free in the liquid, searching for a rabbit antibody to hold on to. Next, we add our second ingredient, the specific rabbit antibody designed to catch cAMP. As soon as we pour this in, the goat anchors on the floor snap shut on the rabbit antibodies. Now, instead of empty chairs, I want you to imagine the floor is covered in a ghost traps. Like in a Ghostbusters movie. The traps are open. They are anchored to the floor so they can't float away. And they are waiting for exactly one thing to wander inside. The ghost. This is where the competition begins. We have two players floating around, and both of them fit perfectly inside our traps. Player 1 is the ghost. This is the real cAMP from my patient's platelets. It is invisible, and we don't know how much of it is there. That is exactly what we're trying to find out. Player 2 is the beacon. This is the fake cAMP molecule from the kit. It looks just like the ghost, so the trap cannot tell the difference. But it has a special enzyme attached to it, alkaline phosphatase. When we mix them together, it becomes a race. The traps are open, but there is a limited number of them. Once a trap snaps shut on a molecule, it stays shut. If I have a lot of ghosts, real cAMP, they will swarm the traps. They will occupy every single spot, and the beacons will be left floating with nowhere to go. When we wash the plate, the beacons go down the drain. But if I have very few ghosts, the beacons will win the race. They will get caught in the traps and stay there. This leads to the golden rule of competitive ELISA. The stronger the signal, the less real molecule you have. It is an inverse relationship. If the trap is glowing yellow, it means a beacon got caught, which means the ghost wasn't there to stop it. Now let's look at the workflow. It starts with the blood collection. I draw the blood into a tube, but I immediately add something crucial: 20 microliters of drug called Tirofiban. Why Tirofiban? Well, look at this platelet. On the surface, there is a receptor called glycoprotein 2B3A. Think of it as the Velcro that platelets use to stick to each other and form clots. Tirofiban blocks this receptor. We need this because the vibration from the car ride to the lab could cause the platelets to activate and clump together, and if they activate now, their internal signaling changes, and my baseline cAMP levels would be totally wrong. We need them to stay in the resting state. Next, I centrifuge the blood at 1500 RPMs for 8 minutes. This forces heavy red blood cells to the bottom, leaving the lighter platelet-rich plasma as the supernatant layer on top. I carefully transfer the PRP from the tubes and pull it all into a single Falcon tube. At this stage, I need to know the concentration, so I take a sample for a cell count. I usually take 5 microliters of PRP and dilute it with 95 microliters of saline or HEPAs, mixing it very gently to avoid activating the platelets. Next, I grab my Neubauer improved counting chamber and I make sure it's perfectly clean. I place the cover slip and pipette 10 microliters of the diluted PRP into the chamber's edge. Then I select the 40x objective on the microscope and start counting. I always follow the L rule to avoid double counting cells on the borders. Once the counting is done, I use the formula to calculate exactly how many platelets I have per milliliter of PRP. You might ask, why count them now? Well, I need to know the total number of platelets so I can calculate exactly how much buffer to add later. This ensures that the final concentration of platelets is identical in every single experiment I run, whether it is today, next week, or next month. This standardization is critical because it allows me to reliably compare data across different days. Once the count is done, I spin the platelets down at 4600 RPM for 1 minute. Then I remove the plasma and resuspend them in Krebs buffer. You will hear about Krebs buffer a lot in pharmacology. It is a carefully designed life support system. Let's break down the ingredients to understand the why. Sodium chloride and potassium chloride mimic the natural saltiness of human blood to maintain isotonicity so the cells don't burst. Bicarbonate and phosphate act as the buffer to keep the pH strictly around 7.4. Glucose? Well, glucose is like lunch for these platelets. They need fuel to run their pumps and synthesize cAMP. Magnesium sulfate. Well, this is the MVP. The enzyme that creates cAMP, adenylyl cyclase, cannot work without magnesium. If I forgot this ingredient, the platelets physically couldn't make the signal I'm trying to measure. Now comes the actual pharmacology. I have aliquoted my platelets into 18 different Eppendorf tubes. This allows me to test the whole matrix of conditions. Some get homocysteine, some get forskolines, and some get prostaglandins. But here is the challenge. Timing is everything. Each tube needs to incubate with the drug for exactly 1 minute. Not 50 seconds, not 70 seconds, 1 minute. Because I have 18 tubes, I cannot do them one by one. It would take forever. And the platelets would degrade. So we have to stagger them. This is a two-person job. I cannot do this alone with just 2 hands. So my lab mate helps me. We work in perfect synchronization. I add the drug to a new tube every 30 seconds. And my friend watches the timer. Exactly 1 minute after I treat the tube, he steps in to stop the reaction. It looks like this. At the beginning of time, 0 minutes, 0 seconds, I treat tube 1. At 30 seconds, I treat tube 2. At 1 minute, I treat tube 3, but at this exact same second, my partner adds the termination buffer to the tube 1. At 1 minute 30, I treat tube 4, and he stops the tube number 2. It is a high-speed assembly line. The termination buffer he is adding is the mix of Triton X100 and acid. Triton is a detergent that pops the cells open immediately. And the acid stabilizes the cAMP so it doesn't break down. Now the cells are popped, but we need to make sure that every last bit of cAMP is released. So we switch the water bath to ultrasound mode and leave the samples there for 10 minutes at 37 degrees Celsius. Next, we need to separate the treasure from the trash. I spin the tubes at centrifuge at 12,000 RPM for 2 minutes. This forces all the heavy cell membranes and debris to the bottom. What remains at the top is clear liquid called the supernatant. This is what contains our ghosts. I carefully collect this liquid, and now we are finally ready for the ELISA plate. Well, almost ready. Before loading the plate, we have to supercharge our samples. We add acetylating reagent to every tube. 10 microliters of acetylating reagent for each 200 microliters of a sample. This chemical modification makes the cAMP stickier to the antibody. And it increases the sensitivity by nearly 20 times. Now we load the plate. First, I pipette my samples and my standards into the wells. Next, I add the conjugate. This is the fake cAMP or the beacon. Finally, I add the rabbit antibody. These are the traps. I put the plate on a shaker for 2 hours. Right now, inside those tiny wells, the invisible race is happening. The ghosts and the beacons are fighting to get caught in the traps. After washing away the losers of the race, I add 200 microliters of the substrate, PNPP, to every well. This liquid starts out clear, but it is chemically programmed to turn yellow if it touches the enzyme on the beacon, the fake cAMP. But the reaction isn't instant. I have to let the plate shake at room temperature for 1 hour and a half. During this time, the color slowly develops. The more beacons that got trapped, the yellower the liquid becomes. Once the timing is up, I need to log that color in. I add 50 microliters of stop solution to every well. This holds the chemical reaction immediately, ensuring the color doesn't keep changing while we are handling the plate. We immediately take this plate to the Hydex machine. This is a spectrophotometer. We set it to measure at 405 nanometers. Now you might be wondering, why 405 nanometers? That specific number corresponds to violet light. And this is just basic physics. Yellow liquid acts as a filter that absorbs violet light. So think about what the machine is seeing. If it looks at bright yellow well, that liquid blocks the violet light from getting through. That tells us the beacons are still there. They won the race. And if the fake beacons won, it means there were almost no ghosts to stop them. So, a strong yellow color actually means that my patient's platelets had very low cAMP. On the other hand, if the well is clear, the violet light shoots straight through to the sensor. That tells us ghosts, the real cAMP, flooded the traps and kicked the beacons out. So, a clear result is actually the jackpot. It proves we have high cAMP. It is an inverse relationship. And that is the beauty of competitive ELISA. By measuring the fake signal, we calculate the real messenger. For my dissertation, I am looking to see if homocysteine changes this signal. If it does, it might explain how it damages the cardiovascular system. I hope this walkthrough helped you visualize the invisible machinery of life. If you are interested in the specific antiplatelet drugs I mentioned, like Tirofiban, I will be uploading a detailed lecture on that soon. Thanks for watching, and I'll see you in the next lecture.